The biotic factors of an environment are all organisms found within the environment. A group of organisms of the same species found within the same environment is called a population. Population of different species sharing the same area and interacting with each other make up a community. One way of studying the interactions of organisms in a community is by taking an inventory of all the species in the area and comparing the sizes of their populations. In this practical, you will be exposed with a simple concept of quadrat. Students also need to observe the abiotic factors of a study site and classify the species of plants in that site. The objective of this experiment are: first, to introduce the simple concept of quadrat. Second, to enhance the power of observation. Third, to record the difference in plant distribution within the quadrat. And the last one, to infer from natural observation the possible factors that affect distribution and abundance of organisms.
So here are our materials. First of all, raffia string. Second, measurement tape. Third, stick or branch to tie our raffia string. Third, scissors. And the last one, your own notebook to take the data.
First, a random forest near the area lab will be chosen as our study place. Second, a 5 x 5 meter quadrat sample will form throughout the forest using raffia string and measurement tape at a random place that have wide variety of species. Step 3, we repeat the same step in step 2 to form another 4 quadrants, since we need 5 quadrants to complete our experiment. Step 4, since our quadrant sample have been formed, now we must find species that are still standing and counted them as many as we can find. And for the last step, we gather in the lab to combine our data together.
Now for the discussion. Quadrat is a piece of equipment that is used to identify a sample area and study immobile biotic organisms. It is used to estimate the abundance and distribution of a species. In this practical, we use quadrat stratified sampling. From the data, we observe that there are species with low abundance and some other species have high abundance. Species Y has the highest individual count and it occurs in 3 out of 5 quadrants that have been studied. The factor that affect this distribution of plant is due to the biotic factors such as water, nutrients, and sunlight. Plants that possess fibrous roots will have a higher count as they have more surface area on the root to absorb more water and nutrients. Tall and short plants will compete for sunlight, which is important for photosynthesis and growth. Tall plants will have the access to get the sunlight first, as it shades the shorter plant. Lack of sunlight may cause it to be unable to survive.
After conducting an analysis of the plant distribution within the quadrant, significant difference or variance were observed. This suggests that the distribution of the plant species within the studied area is not uniform. Instead, there are noticeable differences in the arrangement, abundance, or type of plant present. Next, the spatial heterogeneity of each species in the studied area is due to different method of seed dispersal. Plants that depend on the wind dispersed seed will cause it to disperse in a large area that leads to lower count in one quadrant. On the other hand, plants that depend on the gravity dispersed seed will have a high count as the seed fall near the parent plant. Based on the discussion, the simple concept of quadrant known as it is used to estimate the abundance and distribution of a species. The observation shows that species Y have the highest individual count. After the analysis, it shows that the distribution of plant species are not uniform. The distribution and abundance of organisms can be affected by how their seed disperse. Some plants disperse their seed by wind, and some depend on the gravity. A quadrant is a tool used in ecology and environmental science for sampling and studying vegetation or organism in particular area. It is typically a square or rectangular frame of a standardized size, often made of metal or PVC, which is placed randomly or systematically in a study area. But in this practice, we build 5 by 5 meter quadrant by using raffia string and measurement tape that were formed throughout the forest, which has a wide variety of plants. The distribution and abundance of organisms in the area will be determined by counting and identifying each species present in the quadrant. The two basic requirements of quadrant technique are standardization and randomization or systematization. For standardization, each quadrant used needs to have fixed size and shape to ensure the consistency in sampling across the study sites. Randomization or systematization is when the quadrats were placed randomly or systematically within the study area to ensure unbiased sampling. Random placement help reduce sampling bias and to make sure that the data collected is representing the entire study area. Systematic placement will follow a predetermined pattern to ensure thorough coverage of the study area.
Okay, now we're moving to the post-lab question number 3. What would be the advantages and disadvantages of using a larger or smaller quadrant to survey the sedentary organism? Okay, now for larger quadrant, the advantages is encompass more organism and habitat variability, providing a more representative sample so that more species that we can study. The disadvantage, more time consuming to survey, especially if the habitat is complex or the organisms are dispersed. Now for the smaller quadrant, the advantage is faster to deploy and survey, more efficient for studies requiring rapid data collection since we can our experiment in a short time. But the disadvantage is higher risk of sampling error with smaller quadrant if the population is sparse or the habitat is heterogeneous. This will make our data small or less and we can get accuracy in the data.
Post-lab question number 4. When dealing with habitat of significant spatial scale, such as large forest area or expansive aquatic environment, estimating average densities of organisms become a challenging task for ecologists due to several key issues. One of the issues is limited accessibilities. Large habitat may have areas that are inaccessible or difficult to reach, which poses challenge in terms of establishing sampling location. This limitation can result in uneven coverage of the habitat and may bias density estimate towards the areas that are more easily accessible. Other issue might be detection probability. In habitats where organisms cannot be directly observed, such as dense forests or murky water bodies, detecting individuals becomes a significant challenge. Sampling method reliant on visual observation may underestimate densities due to misdetection. The last issue concerned ecologists is temporal variation. Densities of organisms in large habitats may vary over time due to seasonal change, migration pattern, or other ecological dynamics. Conducting repeated sampling effort over multiple time points becomes essential to account for this temporal variation and obtain more accurate estimate to average densities. There are differences in the number of plant species in the quadrant because of some disturbance event that is caused by fire, grazing, or human activity. The disturbance may have affected certain quadrant more than the others, leading to the difference in plant species dispersal. Some species may be more resilient to disturbance, while the others may require better conditions to survive.